So what I'm going to talk about today is a portion of my dissertation, and I'm going to also put that into context of the bigger picture of my dissertation. And so I'm going to start by talking about parasites. Here's your graphic image warning that hopefully your breakfast is all digested. So parasitism is pervasive across the tree of life. As Jeremy has already mentioned, it's a really important aspect of biology, and I'll say that sometimes it's often overlooked. Parasites have been proposed to play a crucial role in things such as the maintenance of sexual reproduction. It has been proposed as a driver for diversification, and we're starting to think that it's playing more and more thinking that it plays an important role in the maintenance and development of functioning immune systems. Yet, despite this very common interaction that we have, not only it's very broadly geographically distributed and across the tree of life, there's also high diversity in parasites, yet relatively little is understood about parasite history and evolution. Parasite isn't even really a taxonomic term. It's a way that we describe a mode of obtaining resources. And it's very hard to generalize things about parasitism across multiple different host parasite systems. One of the exceptions to this is that when we talk about parasites, we often talk about things like generalists and specialists. But this is a very binary description that doesn't really accurately capture the entire spectrum of host-parasite interactions. One of the better known, at least probably here, interactions in hosts and parasites is the pocket gopher and chewing louse system that has been worked on by some mammalogists in particular. And it's demonstrated this pattern of codiversification and co-speciation. And so that can be really important in driving parasite diversification. But we also know that there are other things that play an important role in parasite diversification, such as things like climate and host history. So what I'm going to talk about today is how we can test the importance of these different factors in driving diversification in parasites. And with this aim of finding out if it's even possible to identify processes that are common across parasites, or is each interaction really a unique setup and something that we can't really duplicate through different systems. To do this, I propose that we need a system that is geographically widespread, and it has parasites that are inhabiting similar niches. So in some ways, they're ecological or evolutionary replicates of one another, and these are in the same host populations. I will posit that I have a great system for that, which is chipmunks. So I am mostly going to be talking about Western North American chipmunks today. They are currently Tamias, but it has been recently proposed to elevate them to Neotamias and bump up the 3 subgenera. I'm going to refer to them as Tamias today, but keep that in mind. There are 23 species of chipmunk in the Western subgenus in Western North America. They've diversified within the last 4 million years, and they're characterized by this history of divergence with gene flow. So when we look at the phylogeny of chipmunks, of Western chipmunks, this is a time-calibrated phylogeny that was based on 4 nuclear loci, and the stars there are representing the branches that have support. So with these 4 nuclear loci, they were able to resolve the relationships among most of the chipmunk species, but you can see that it's not actually, there's not high support across all of the branches of the tree. A few other things I want to point out about chipmunk phylogeny that we know right now. One of them is that there's support for this Tamias amenus and amenus cratericus separation. So these are 2 divergent lineages within amenus. So the top one is the amenus baculum in those images, and the bottom one is the amenus cratericus baculum. So they're morphologically and genetically distinct. And Jeff Good's group at Montana is actually working on this right now. Other thing I want to point out is that a lot of these species have been grouped into species groups, and I'm pointing out the Quadrivitatus group and Townsendii group because I'm going to keep coming back to them. First, I'll give you a little more information on the Quadrivitatus group. So this is just a cytochrome B phylogeny for the Quadrivitatus group, and it's color-coded by species. So the 2 species that are circled on the right in black are monophyletic using the cytochrome B, so that's Rufus and Canopes. The other ones, each of those colors all mixed in together represent what we're calling species. So this is just to illustrate that there's been high introgression across these 4 species, and it's geographic. So we know that there are regions where if you have a umbrinus and an umbrinus and a dorsalis together, the individuals you capture, their mitochondrial DNA is not going to sort according to their species identifications. So now to get into a little bit about the parasites of chipmunks. I'm going to talk about two of the parasites today, but I'm going to mostly focus on these lice. So sucking lice are wingless insects. They feed on the blood of the host. They spend their entire life cycle on the host. So they're not actually easily transferred from one host individual to another. And chipmunks are known to have two species of sucking lice. These are actually two species of lice in two different families. So the top one is Hoplopleura, and the bottom one, Neohaematopinus. And then chipmunks also have two species of pinworms. So heteroxynema and Rauschtenaria, which I'm just going to briefly talk about today, but I want to bring them up. When I started working on this, I actually started before I went back to graduate school, and in 2007, this was the status of our knowledge about chipmunk parasites. So out of the 23 species listed on the left, the dots represent whether or not those parasite species had been described from that host, and I decided that was not sufficient knowledge. So working with John Demboski at the Denver Museum of Nature and Science, and then again with Joe and John at the museum in New Mexico, and then also with the collaboration from many people, we have done a lot of field work and a lot of investigation. And this is just the field sites and the specimens that I have personally collected, but we have also had many people send us specimens. Duke Rogers, Eric Rickert, and Jim Patton, and Jeff Bradley have all sent me specimens as well. So we've tried to take a pretty thorough survey of the parasites of chipmunks. And in addition to the field work, I also had an undergraduate working with me, Diego, who combed over 1,000 museum study skins. So we were actually able to retrieve lice from museum study skins, and we retrieved them from skins from 1897. However, we didn't try to pursue genomic work with those specimens that old. I also was able to get some middle school students involved in this project, and I took a bunch of study skins to a school, and they all combed chipmunks and pulled lice off of them for me as well. So this is fun outreach. But so this is what we know now. So after this exhaustive survey, I have actually been able to greatly fill in our knowledge for what we know about chipmunk parasites. I'll note that several of the empty spots on the pinworms are because I haven't examined those, well, I don't know if it's because, but I haven't examined those host species. We were able to get complete sampling for all the chipmunks because we use these museum study skins, which added a lot of information for us. So just to give you a little bit different perspective on this, just throughout this work, we've been able to add our known host species for each of these parasite species by, I mean, just tremendous amounts. We've more than doubled a lot of them. And so this is just kind of, I want to throw out my museum pitch that one, museum specimens are really important for a lot of things, and sometimes it's unforeseen, but also that parasitism is this great realm of discovery that is just really underappreciated. You can make a lot of advances just by going out and looking. So overall, my research is kind of investigating some of these broader questions. So how parasites are diversifying with respect to their hosts. Are there signals of codiversification across parasite species? But also, can we identify processes or conditions that are dictating codivergence or host switching across these parasite species? Can we identify these similar patterns that may be able to lead us to similar processes? I'm going to talk for a moment about what has been done with the pinworms. And to do this, I have to acknowledge an undergraduate who also spent some time working with me. Kendall came and spent a summer. He was an undergraduate student at Berkeley, and he spent a summer working with me in the lab in New Mexico on this pinworm project. So we have just sequenced mitochondrial and a few nuclear loci for the pinworms. But what we found is interesting. This is a Bucky concordance tree. So this is a tree just of the mitochondrial marker. And what this does is that it looks for concordance across the lineages between the two pinworm species. So this is one tree illustrating concordance between two pinworm species. And you can see that that purple star is denoting at least half of the lineages are concordant for these host-associated groups. So each of those color-coded lineages corresponds to a host-associated group. And this is concordant across the parasites at these deeper levels. However, if we zoom in a little bit and we look at the tips on these trees, they're not concordant at the shallower levels. So these two pinworms seem to have experienced similar processes that are shaping these deep divergences, but the shallower histories are different. And I think that some of this is being driven by different host contact histories and different things going on at the landscape scale. So coming back to these questions, now I'm going to talk a little bit more, focusing in on the lice. So given, again, this situation of similar host niches and shared host associations, I'm going to try to untangle what is driving host or parasite diversification in this system. First, I want to point out that other project with the pinworms was great. I was able to get really great sampling. I had over 400 louse samples, or pinworm samples that I used in that project. But here I wanted to do a genomic approach, which means I got to greatly increase my sampling at the genomic level for all of these specimens. But that meant I had to decrease my individual and population level sampling. So there's still some questions yet to be dug into with this. When looking at how parasites have diversified with respect to their hosts, one of the ways to do this is to do a tanglegram. So this is just the phylogeny of the host on the left and the phylogeny of the louse on the right. This is just for illustrative purposes there. And we predict to see some levels of codivergence. So we expect to see at least some codiversification between the hosts and parasites. So here you can see that although not all lineages are codiversifying, you can see that there would be some examples where sister hosts have sister louse lineages. We also predict there to be signals of geographic structure, and this is partially based on our findings with the pinworms. We do think that there should be some geographic scale processes that are dictating parasite divergences. We also predict that the two louse species are going to have different phylogenies relative to their hosts. So this is putting it into the context of how are the parasites interacting with the hosts differently or similarly. We're predicting it's going to be different because they're in different families and they've likely had different histories with the hosts. We also did some chipmunk sampling for this. So we had 63 individuals representing all 23 species of Neotamias, and because they're my focal group, I included Sibiricus and Striatus as outgroups. And then for the lice, we had 35 Hoplopleura, arboricola from 19 host species, and we also included one of the Tamias striatus lice. So striatus is the eastern chipmunk, and it has a Hoplopleura louse, which is Hoplopleura erratica. For Neohaematopinus, we didn't have a good outgroup for this, so we just included the Neohaematopinus as our focus. And these lice included samples from museum study skins from 1937 that Chester Lamb actually collected in Mexico. So that's from the yellow arrows pointing out a Tamias derangi collected from Mexico in 1937 by Chester Lamb for the Moore collection. We also had other samples from 1957, 1997, and 2001. So this is an individual louse that was ground up. And so on the bottom here, I just want to hope you can get a little appreciation of the size of these lice. So we used one louse to do this. And we did whole genome sequencing for the lice. We just did shotgun whole genome sequencing. They have relatively small genomes, which means we were able to do this economically and efficiently. Then we took 1,106 one-to-one orthologous loci that had been identified from the human sucking louse, and we did targeted assembly of those using the software ATRAM. For the chipmunks, because we're looking at a different scale of a genome here and resources, we used ultraconserved elements. So we did ultraconserved element sequencing with rapid genomics, but then we used ATRAM also to assemble those loci. I just want to go ahead and say now, the UCE data are very new, so I'm still looking through these. The RAXML gene trees were generated for each locus with 100 bootstraps, and then we did use those maximum likelihood trees as the input for Astral to get species trees. We had 100 bootstrap replicates in Astral for the lice. I ran out of time to get 100 for the chipmunks, so what I'm showing you today for the chipmunks only have 20 bootstraps in Astral. Just to quickly go over what we were able to get from the lice. So we were able, out of our 1,106 target loci, we were able to assemble 432 loci for all of the samples. But if we removed that one louse sample from 1937, we were able to assemble 975 loci. For the Neohaematopinus, we got almost 900 for all 23 samples. And then for Tamias, it's a different scale. We got 160 UCE loci for all of the Tamias samples. And we got about 3,300 for half of our total target taxa. So this is the Tamias species tree. This is complete taxon sampling for all currently recognized species of chipmunks. And I just want to point out a few things here. So the first thing is we were not actually able to resolve the relationships within the Townsendii group. The Sonomi, Speciosus, and Senex that are pointed out here, we did not recover monophyletic relationships for those species. Oh, and also just note that there are very high bootstrap supports on these branches. So these are well-supported relationships with the loci that we used. Also want to point out that the red box there is around Tamias minimus, but the red arrow is pointing to a minimus that is not within that group. Those are Tamias minimus grisescens, which are from Wyoming, and they have also been previously identified as a unique lineage that's probably worthy of elevation. And then another one that came out not where I expected it was that Aminus. So I thought that this one individual of Aminus that was not with the rest of the Aminus was probably Aminus cratericus, but we looked at the baculum and it has a standard Aminus baculum. So we think that this is a very complicated system, and we're not going to try to draw conclusions about that right now, but hopefully we'll learn more soon. And then finally, Tamias palmeri and umbrinus were monophyletic. Other people have proposed that palmeri is not actually a distinct species, that it is just an isolated population of umbrinus. And what our results with the UCEs are consistent with that. So moving on to the lice. So this is Hoplopleura, the tree on the left, color-coded with the dots on the right just to show the localities. These phylogenies were made with the Neohaematopinus, so the other species of louse, as included as an outgroup, but then after we made the species tree, we pruned that individual and did midpoint rooting. And when we did that, the striatus louse actually came out as an in-group. So the Hoplopleura arboricola or the Neotamias lice are not they're not a monophyletic group with respect to the striatus louse. And if we just look at the overall raw pairwise sequence divergences between the two Hoplopleura arboricola clades, it's higher than either of them are to the erratica. I just want to point out these are exons, so there's not even introns in here. This is pretty high divergence for exon data. Also, I want to point out this one locality in California where we had a speciosus and an alpinus, and those individuals at the same locality are hosting divergent lineages of lice. And then the quadrivitatus group. The quadrivitatus group lice, so all of the lice collected from those host species, sort geographically. So there appears to be strong geographic structuring shaping the divergences of the lice associated with those host species. And now looking at the Neohaematopinus relationships. So first, that same population in California where we have speciosus and alpinus, they both host the same lineage of louse at that one locality. So right there we can identify this different dynamic going on between the two parasite species. Also, this is the quadrivitatus group. So these are the lice collected from the quadrivitatus group, and they're not monophyletic as their hosts are, but they do have a bit of a geographic signal going on. So now I'm going to zoom out and we're going to look at the tree kind of from a straight comparison with the hosts. So on the left is the chipmunk tree, on the right is the louse tree. This is the Hoplopleura tree. And what I've just pointed out here with the arrows is a few of the points worth noting where I think that these are going to be considered host switches. So that top arrow is pointing to the alpinus louse, which is actually coming out sister to Aminus, but still closely related to minimus, which is what we would expect. And then in the bottom is where we're recovering lice from Panamentinus with the palmeri and umbrinus lice. So zooming in on those, this is looking at the Townsendii group relationships. Each of those stars denotes a pattern of codivergence. So we're not testing the depths or the timing of these relationships with these trees. We're simply observing if the lice that are collected from hosts that are sister are also sister to one another. So at the deep level, the Townsendii group are monophyletic, but once you zoom into that, we're recovering different relationships from the lice than the hosts in most places. When we look at the Aminus, alpinus, and minimus, you can see that there is very little signal of codivergence once you zoom in. So at the deeper scales, it does look like there's some signal of association. And then again, looking at another part of the tree where we just have this mixed pattern where at deep levels there are some signals of codivergence, like for the quadrivitatus group hosts and their lice, but then it's different at varying scales across the tree. So this is looking at the other species of louse, looking at the Neohaematopinus tree. And again, the red arrows are pointing out places that I am going to call host switches. So the top one there is where there is an Aminus louse, or an Aminus host, and the louse was recovered in the same clade as the Townsendii group lice. And then we have the Panamentinus again, and then the alpinus with the speciosus. So looking at the patterns across the tree, we have these deeper signals that appear to be codivergence or a history of association with these hosts. But at the shallower levels, there's some host switching going on among these Now to just kind of walk you through again some of these comparisons I've made between the two parasites. It looks like my species name got carried over there for me. So first, in Hoplopleura, we have a monophyletic group from the Townsendii clade, Townsendii hosts. On the right, we have Neohaematopinus that is mostly monophyletic with Townsendii hosts, but there's also an Aminus host in there. And then we have the Aminus, Alpinus, and Minimus lice being sister to one, or forming a clade for Hoplopleura, but not for Neohaematopinus. And then we have the Speciosus obscurus and Mariami groups looking different between the two species. And then again, this last one, the Quadrivitatus group hosts. So the Quadrivitatus group hosts for Hoplopleura are monophyletic and not for Neohaematopinus. So to kind of summarize what we found with the lice, the question as far as how each louse has diversified with respect to the hosts, and if there is a signal of codiversification, these lineages correspond to host groups. So this signal does appear, especially for the Hoplopleura. They've been associated with these hosts for a long time. There's a deep relationship going on here, but we're not prepared to say it's driving codiversification. This could be more of an instance of isolation. And with the Neohaematopinus, there's actually less correspondence of host lineages and host groups. And this structure also does not suggest codiversification. And then when we look at comparing between the two species of lice, it does appear that both have this geographic structure shaped by the landscape as well as their host associations, but we think that they're being shaped in different ways. For example, where the Quadrivitatus group hosts are monophyletic for one and not the other. And we also have identified what we're calling more instances of lineage host switching in the other louse species. So to kind of summarize overall with the chipmunk parasites, Hoplopleura is a much more common louse. That's the louse we find more often. The clades do largely correspond to the host groups, but the relationships among those clades are different. And we've only identified 2 host switches, but granted, we have very relatively small sampling here. Whereas with the Neohaematopinus, which is a much less common louse, it has less correspondence of clades in the host groups, and we have identified relatively higher rates of host switching in this group. So for now, it appears that Neohaematopinus has a shallower history of association with chipmunks than Hoplopleura. And just put that into context with the pinworms. The one species of pinworm, Heteroxenema, is a much more common pinworm. The clades there do correspond to the host groups, and we identified 7 host switches out of 224 samples we looked at. Whereas the other species of pinworm, the Rauchtineria, is again the less common pinworm. The clades do also correspond to the host groups, but we identified a higher rate of host switching. And we think that this is because Rauchtineria has been associated with chipmunks for less time. It's a more recent parasite of chipmunks. So to zoom out a little bit and talk about this as far as just parasites in general, we see this deep structure of the sucking lice and pinworms with these host-associated lineages. And these between the 2 pinworm species, they have similar relationships between the host-associated lineages, but we think it's different timing. There are deeper divergences in one species than the other. The relationships among the lice host-associated lineages are not only different from one another, they're also different from the pinworms. So we're not finding a generalized pattern of diversification across all of the parasites. All of the parasites that we examined have at least some amount of host switching among the lineages. So we've identified these lineages where all of the parasites recovered are primarily from one group or one closely related host group or host species. However, we do occasionally find that lineage from a different host. Future directions with this. So one of the great things about doing the whole genome sequencing for the lice, that now means that we have this just tremendous data set of lice data. And we would like to look for selection in these lice. And we'd also like to date the divergences. So we think that by characterizing these interactions at the genomic level, we can hopefully get a little closer to the ideas of truly testing for coevolution. And we'd also like to generate time-calibrated trees for the lice. And by doing that, we can test if the timing of diversification corresponds to chipmunk diversification events or climate. We suspect it will correspond to both of these things. And finally, just to the significance of these types of projects are that, I'm just going to say largely, parasites are really understudied. Parasite diversity and distributions are virtually unknown for most wild mammals, yet I think that they're really important and they are key for a holistic approach to ecology and evolution. Parasite diversification is complex. It is impacted by host association, history with the hosts, host demographic, and then how landscape and climate have played into shaping all of these things as well. With that, I have a lot of people to thank, not only the collaborators that I've worked with on this project at UNM, Denver Museum, and University of Illinois. I have a lot of funding to thank, particularly the Society, not only for the fellowship, but also for the grants and aid and the travel awards I've received throughout the years. And all of the people who have given me samples or helped me out with fieldwork for this project. It's been a tremendous collaborative effort just to get the data collected. And all of the museums for the samples, that's a really important part of this work as well. And with that, I'll take questions.
Do you have time for a couple questions? Questions for Kayce?
Nate.
Yeah.
In regards to the UCE data, I'm curious about how much variation you were able to get on the scale of Tamias, and then how that compares to the Sullivan phylogeny.
Yeah, so the question was about the variation in the UCEs and how it compares to the Sullivan phylogeny. I can't really answer it that well right now because I feel like those trees were a first go at it. If I just blindly looking at it, there's not a ton of variation in the UCEs. And the fact that we were only able to recover 160 loci for all of our specimens isn't too promising at this moment. Overall, the relationships are similar to the Sullivan phylogeny, the exceptions being the Amenus and Ruficatus relationships. I think there was another one I can't remember off the top of my head. But so it's a little bit different, but also similar in a lot of ways.
All kinds. Bruce.
You identified a number of cases of host switches within this relationship. I wonder if you could place that in the context of the distributions of the chipmunk species involved.
That's a great question and something I should have pointed out. So Bruce's question was if I can place these host switches in the context of the distributions, which is really important. Yes, so all of the host switches that we have identified are where host species are currently sympatric. One of our hopes was that we could test past contact and actually identify past host species contact with host switches, but we can't because all of the host switches we've identified thus far are with currently sympatric species.
So one more. Laurel, did you have your hand up?
Yeah, we kind of answered it. I was wondering, it's like for some of the older species from the 1800s, are you still getting the same, like, is that relationship persisted of the lice and that?
So the oldest sample that I have the genomic data for is from 1937, and so that was exactly as we would have expected. That dorsalis, so that came from a dorsalis specimen, and that dorsalis louse fell out in the phylogeny with the other regional dorsalis specimens.
One more. One last.
Are there other parasites that you can study, like trematodes?
So there are other parasites of chipmunks. These are by far the most common. I think I should know these numbers off the top of my head. I find pinworms about 60% of the time and lice a little bit more often than that. Every once in a while we'll find a tapeworm or a different nematode or an acanthocephalon, but by and large it's pinworms and lice on these. There are also fleas, which Helen and John Piggage are here somewhere, and they are working on the fleas as well. So they have a great assemblage of parasites, but the lice and pinworms are much more common.